Yeah, okay. So I'm going to present to you today medical image analysis and machine learning. Just to introduce the topic, in a past presentation, some of my colleagues presented to you image processing in general. So today I want to introduce you to image processing applied in medical images with a couple of real examples and real application. So the first one is a new tool for predicting the secondary structure of the protein based in decision trees. In this application, we propose a method based on artificial intelligence and mixture of experts. These words present a new tool for predicting the secondary structure of the protein called PESPAT, which significantly exceeds the accuracy of predictive methods available now. So this tool is based on model constructed by decision trees of the mixture of experts. The second one is an histological image bank of human cardiovascular system called VSCAR. VSCAR is a wet histological image bank of whole sample of the human cardiovascular system. This is a kind of microscopic images. So VSCAR is based on healthy histological sample. For that reason, it's more focused on teaching and learning purpose. And it is composed of different stages. For example, this complete development, this complete method includes sample preparation, image capture protocol, storage structure definition, vocabulary definition, information management tool development, and also a web resource availability. This one is an automatic classification of histological images and histological knowledge model of the human cardiovascular system. The idea in this work is to mix different kind of input source. In this case, we have 2D images that is also histological images. But we also had histological knowledge and expert knowledge. So this work is about how to use different types of resource in the same way to obtain a more complete and accurate knowledge. This proposal enables us to obtain more consistent information because we are using more input source. To reduce margins of errors as well through corroboration and verification of the final classification. This proposal was comparing with the use of every data source individually, and the accuracy was improved a lot. So in this case, we propose to use texture descriptors. That is a kind of information that we could get from the images because the color information is limited to the extent used in this kind for this specific problem. So then in this proposal, we use also more generalized and after that, we apply machine learning techniques to classify and identify tissues, some kind of cells, and also organs present in this kind of images. So on the other hand, we have the modeling using an ontology. This ontology is a way to model the human knowledge in a way that the machines could understand it. And And that is useful to communicate specifically, clearly, and precise concepts. In this case, histological concepts. So also we could represent or model knowledge of humans, and we could make interaction between humans and machine in an easy way. Finally, with an ontology, we could make induction or reasoner to obtain new knowledge applying logical mathematical. So this is a very powerful way to use the machine computing. And at the end of this proposal, we'd refine a process based on the image classification, mixing with the histological ontology. And this final refinement allow us to identify not new, but more structures present in these images which weren't identified in the automatic classification. Like cells, fibers, or even know to what system that organs is belong. This is other kind of application of image processing in medical area. This is called new biomarkers in pathological image for breast cancer. The aim was to identify new biomarkers to know what is the hormone resistant in this breast, in this topic for the different patients. The idea, you could see here 3 different images: respond, no respond, and recidive. These are the 3 different states of a cancer patients. And the main of this research work was to identify the nuclear and cytoplasmatic biomarkers. As you can see, the first image is just like they have stain in the nuclear cells, that is small balls that you can see in brown. And the second one is cytoplasmatic because they are staining the rest of the tissue except the nuclear or the small balls. The main of this work was identification, quantification, and finally has some statistics about that. This is another sample. In this part, we are trying to predict the risk of recurrence of early stage breast cancer patients. Using, again, different kind of information together. In this case, we will have signature score, pathological data, clinical data, and also environmental or lifestyle risk factors to get the risk of recurrence for each patient. The idea is to have a more personalized prediction. We propose to use deep learning solution. However, according to the number of data sets that we have available. So we use transfer learning techniques, which is a way to use deep learning in a better way because we don't need a lot of images. And in this proposal, we will use clinical information, genetic information, and also the signatures about what is the risk of recurrence for each patient based on the genetic information. And the output will be the risk of recurrence for each patient in 2 categories: high risk or low risk. There are a couple of more examples. In this case, they are using MRI and CT images. This is another totally different kind of images. This project is called in silico biomarker for epilepsy surgery. This is the first aim of this project, which is create a Colombian brain MRI atlas, because each region has different anatomical characteristics. So this is a very important aim, very important project. The proposal is to make anonymization the first step. This is mandatory for all these kind of projects. The second one will be pre-processing and made an average to get a brain atlas. The second object of this research project is to make a multimodal medical image fusion in stereotactile electroencephalography. The idea in this one is to have two different kind of images, the CT and MRI. There are two different kind of medical tests to extract the electrode node segmentation, which you could see in the upper middle of the images. There is some artifact that the physician used to have a guide. And also our aim is to fuse the images to get a more complete analysis, because with CT you have some kind of information and with MRI you have another kind of information. And the last objective of this project is rendering post neurosurgery samples. The idea here is to extract this. For example, in this case, we are talking about tumor. When the physician extracts the tumor, so that tumor loses their shape, size, and other characteristics so quickly due to the environmental parameters. So the idea here is to don't lose that very important information with an in silico sample. So the proposal is to have a pre-surgical image and also a post-surgical image, and mixing both information, have a registered or it's like a kind of fused image to locate the sample. With this kind of identification, so we will keep all the characteristic about that tumor or that sample, like a shape, like a size, and other very important information to analyze what is the case of the patient. And that's it. So if you...
Oh, okay, great. That's brilliant. Thanks a million, Claudia. If you could just unmute anybody there, and if anyone has a question, you can certainly go for it now.
Done.
Thanks a million. Does anyone have a question for Claudia?
That's fine.
Claudia.
Ricardo.
That's Ricardo. Yeah, how are you doing, Claudia? Good morning.
Fine.
So quick question there, when you're saying you detect, the organs there, do you use pre-trained models there, or did you get the images, from some sort of database?
Which one? This one?
Yeah, in general, when you were just saying that, you detect different kind of organs. So do you use like pre-trained models on transfer learning, or you just went and trained your own models?
Yeah, I present many of different cases. In some of them we are using, we train a new model. But for example, in this one, we use pre-trained models because we are using transfer learning, that is try to use pre-trained models to reduce the number of images in this stage of the retrain steps.
Okay, so I presume there's loads of available models there just have been pre-trained with different organs, right? Loads of organs.
Yes and no. We are using pre-trained models that were trained with different kind of images, but have good result for medical purpose. But for example, in this one, we just use SVM solutions. So we didn't use pre-training model. In this one was SVM cascade SVM.
Yeah.
In this one were decision trees. So we have different examples.
Very good. So yes, last question was, so when you're training from scratch, if you have done that there with some models, how many images did you use? I'm just curious there.
In this case, let me see. In this case, we have more than 8,000 images per class, but we are using block, like a block packets or small blocks. So there are no the complete sample. Let me see. In this one, we have a reduced number of images. I don't remember exactly the name, the number, sorry. In this case, we had more than 6,000 blocks per classes, but we are trying to get more. And about CT and MRI is more complex because for each patient we only have 2 or 4 images. So we are still trying to get more samples in this case.
That's very good. Thank you very much, Claudia. Amazing.